is due to the heterogeneity of these tumors, which people have recognized for quite some time. When we talk about heterogeneity, we can mean a lot of different things at the genomic, transcriptomic, proteomic level. We can mean spatially and temporally heterogeneous, and then we can mean from patient to patient, obviously, as well. My mentor pictured there during my residency was Anup Patel, who was out here at UW and has now moved on to Duke. And he was first author of this seminal paper for single-cell RNA sequencing for GBM, where he really deeply profiled a series of tumors at the transcriptional level. And instead of doing bulk sequencing, did cell by cell and identified, four major groups of tumors and showed significant differences in their overall behavior and survival. Since then, we've kind of come a long way in identifying more states. At last year's Society for Neuro-Oncology meeting, Mario Suvo is presenting work where he proposed up to 13 different transcriptional profiles for GBM. So we're really recognizing that this is, a fairly complex and heterogeneous disease entity that's really going to require some very specific tailoring. And given that genomic complexity, it's pretty unlikely that we're going to be able to treat it effectively with a single agent. So the question is there some specific combination of agents that would make sense for a regimen? And that's a little bit of what the general thrust of this paper was about. So I'm going to turn it over to Annie to talk us through some of the results from the paper here.
So the rationale to do this meta-analysis is based on the hypothesis, can we find a treatment for a specific tumor locations or a combinational treatment for all GBM? So in any ways, we can slow down the tumor progression, and hopefully we have enough time to figure out patient-specific treatments. And so we integrate the MRI imaging data with RNA-seq and proteomic data. Then this way we can find the common and unique molecular signature. Then we apply those signature into the computational search and to identify the therapeutic compound. Next slide. So this string F interaction analysis kind of summarize the top pathway commonly upregulated in all GBM, and each circle represents a genes involved in different pathway. And so we upload those genes along with the pathways into the computational program. The next slide. Those computational programs include the evolutionary algorithm and the PubChem database, and utilize those to search for the candidate compounds. And this scheme summarize the compound identified from this computational search. And for example, and also it highlight the drug activity. So for example, the benzimidazole, that is the azole drug, and is known in the antimitotic drug. And also we found the endoperoxide containing compound is from the big sesquiterpenoid families, and those family has known function to inhibit cell cycle progression, such as blocking the G1S transition, and also some of those compounds has a function to promote the cell death pathway. So we test all those candidate compounds. I think it's the next slide. So we're using those primary GBM cell and also GBM-derived tumor sphere to test those compounds. And the preliminary test is showing the benzimidazole has kind of moderate effect, but the dihydroadenosine, DHA, is belong to the endoperoxide containing compound. This one has a greater cytotoxicity against GBM cell at a lower concentration for about 48 hours treatment. And so now the next one. So then we also look into the distinct gene set associated with different tumor locations. So this heat map summarizes a distinct gene set those are differentially changed, and the change is associated with tumor location. So, for example, the PLK1, the polo-like kinase 1, is increased in the group 3, so it's cortex-associated GBM. And so now it kind of gives us the idea we can, using the similar approach, put those genes and the pathway into the compound search. But now we know the PLK1 compound library is available. So the next candidate compound testing would be, we can think about combine the anti-mitotic drug with DHA inhibit cell cycle and also the PLK1 inhibitor, particularly for the group 3 cortex-associated GBM. So that's kind of the idea, what's the future direction for the combinational therapy. And now I'm going to hand over to Dr. Emerson, continue on discussion of future direction. Thank you.
There we go. Thanks, Annie. Yeah, so where we want to take this from here is how can we really effectively model GBM in such a way that we could even screen potentially identified combinatorial therapies for specific patients. And this gets a little bit back to this issue of heterogeneity, particularly, at the single cell transcriptional level within a particular tumor. And there's a lot of different factors that generate and sustain that heterogeneity, but one of the big ones is the actual tumor microenvironment and the cytokines and stromal support and things like that that really are key for maintaining that transcriptional diversity. So we wanted to try to see if there was a way we could improve on that microenvironment fidelity to maintain that heterogeneity. And so the idea with that is the use of something called organoids. When people talk about organoids, they can mean one of two different things, and they both get the same term. Some people use it as just a little chunk of tumor tissue that you throw in suspension in a dish. And that's very common in like the colorectal and genitourinary specialties, but some people in GBM use it that way as well. The way we mean it is more a three-dimensional brain-like structure also grown in a dish, but is derived from pluripotent stem cells, either embryonic stem cells or in our case, pluripotent stem cells. And these can be, in some ways quite complex and sophisticated structures that show a lot of the same temporal and spatial patterning as the native brain. People have done things like shine lights at them and shown that photoreceptors get activated. They've even slapped EEGs on them. That being said, these mini brains are still obviously quite far from actual vision or cognition or anything like that. So I want to acknowledge that limitation. But the idea here is that we can take tissue from the operating room, from our GBM patients, and transplant it into these organoids so that we have kind of both types of tissue represented, both malignant and non-malignant cell states. And that way, the idea is we would have a better fidelity of the number and diversity of cell states represented, which could help us with screening our proposed therapies. So these are some nice immunofluorescent images indicating the diversity of cell types that we get, including neurostem cells with SOX2 and PAX6, 4 brain identity markers in FOXG1, neurons with double cortin and MAP2, and then astrocytes with GFAP, oligos with OLIG2, and even microglia with TMM11, 119. And then we've actually started improving the complexity of the model to include vasculature in the form of endothelial cells, just to really try to make these as brain-like as we can in a dish. And we do that and we transplant the GBM tissue, we can see it actually, integrate, start to divide, and even begin to invade within the quote unquote normal brain tissue. And then, some work from Howard Fine a few years ago that where he tried transplanting some glioma stem cells into organoids, showed that those tumors would retain a lot of the genetic aberrations, including EGFR alterations, that those cells would not retain in like standard two-dimensional monolayer cultures. Mario Suva, who I mentioned earlier, performed similar type experiments and showed that we were better able to identify the number and types of cell states in these models compared to conventional gliomospheres or other cancer stem cell in vitro setups. So part of my work with Anoop back in residency was to set up a system to model this kind of screening, where we would profile the tumor at the primary occurrence and then at recurrence, profile them, and then based on computational analysis, be able to select rational drug targets from them. And when we do that, we can really fill in some of the evolutionary trajectory of the disease by profiling at various time points, including immediately post-treatment, late post-treatment, and at the time of recurrence. And you can kind of see some of the shifts in cell states that are represented as we move through that disease. So then the schematic here kind of shows, again, the general idea that we can start to learn more about the various cell populations that are either induced and or selected for, and then choose logical targets to come up with a more complex drug regimen for patients at recurrence, who, as you know, often have no standard option. It's often left to the oncologist's best choice for what to do when the tumor invariably comes back. So that's kind of a broad 30,000-foot view of our work so far and where we hope to take it in the coming years. Annie and I just submitted an R01 last month outlining a lot of our hopes for where we hope to take this in the future. Again, I'm sure a brief overview like this left you with more questions than answers, but I'm happy to take any of those questions now.
Thanks, Sam and Annie. That was fantastic. I'll jump in and start. I think, our lab actually studies this too, and we've a number of years ago we published a paper where we looked at a subset of 5 different GBM cell lines that we radiated, and we looked at just standard RNA-seq on them, and then animal models, and found that every single one of them had a more mesenchymal shift when you looked at the principal component analysis. So I guess it begs the question, how does the standard care really change what you're looking at when your initial genomics or RNA expression, protein expression is one thing, and you're really selecting for something different? And how do you propose to be able to model for that? And I think you touched on that in the end, but from a practical perspective, what's the ideal?
Yeah, I mean, I think it's an astute observation, and you almost have to treat primary and recurrence as 2 completely different entities for the reasons you're saying, because it's a lot like antibiotic resistance. You're selecting for more virulent strains, and as mentioned, you're maybe inducing transient things. I'll say in my own work, I've seen similar shifts with the organoid model towards mesenchymal cells. Similarly, a similar shift in macrophage polarity from more of an M2 to an M1, so more of a pro-inflammatory, which, depending on your view, you might think, well, maybe that's good. Certainly, a lot of people have put in a lot of effort to trying to figure out how to heat up GBM in the first place.
Yeah.
But you're absolutely right. I think it's a totally different animal by the time this tumor has seen surgery and radiation and TMZ. And a lot of times, what we're getting are just at the early stage of the disease and then very late in the course. So I don't know if Annie, if you have anything you want to add in there as well. No. Okay. We'll give her sore throat a rest.
Well, I want to echo Chris's sentiments. Great paper, and it does prompt a lot of questions as we were sort of going through an incredible summary. But how do you think that this approach, and sort of thinking about future directions, this spatial transcriptomics, really might change the way we're classifying GBMs as sort of the discrete 1 through 4 classification, using just purely MR, or even thinking about more sophisticated MR like MR spect or MR perfusion? I mean, this to me seems to open up the possibility that these discrete classifications are really just as overly simplified way of looking at things, and the way of the future might be more of this transcriptomic type approach like you've outlined. And then one additional question is really just about the the therapeutics that you're thinking about, are you also thinking about or prioritizing the potential relative blood-brain barrier penetration of those agents? Is that part of the process that you think would be important, either that these be small molecules or other sorts of things that might get across the BBB, or that you might even consider using different methods? And we've talked about this in past tumor talks, using things like focus ultrasound to open up the BBB. But
Yeah.
I know those are sort of two combined questions, but I'm thinking more kind of around future directions and related to the work that you've put forth, and Dr. Lin as well.
Yeah. Annie, did you want to go ahead?
Yeah, so the project, the proposal Sam and I submit is really focused on how we can engineer the delivery vehicles, especially those compounds kind of hydrophobic. So we figure out a way how to encapsulate them and then can deliver them to pass through the blood-brain barrier. And so I think we can talk about that if we have time. Sam can mention what we did for those nano carrier. And
Yeah. I was just gonna say, I mean, yeah, not to, give away the whole proposal, but a large part of it centered around engineering extracellular vesicles for this delivery to get at some of the issues you're talking about. And then as far as the, whether our current classification is a bit too simplistic, I mean, I think in a sense it's crude, right? And there's certainly a lot of grade 4 GBMs. I know that's redundant, that behave very differently. And I do think that, whether it's at the transcriptional level or the epigenetic level, I would love to see this kind of technology improve to the point where it's fast enough that it's something we could really use intraoperatively and help us figure out, well, how aggressive do we need to be? I mean, super maximal resection has been a buzzword now for quite a long time, but it would be good to know how often do we really need to chase all the flare stuff and, make it make sense to guide our strategies in the OR.
And I think you've hit the nail on the head with that. Obviously not all grade 4 tumors are equally alike, and I think this is just one more example of what has historically been, and for the most part, while I was in training, and perhaps while you were too, that we so oftentimes place these as very discrete classification based upon MRI and maybe even histopathology. But spatial transcriptomics is really just, I mean, yet another means of teasing these apart, separating, and obviously providing some therapeutic options that really relate to the underpinnings of that tumor. We have a question from the audience, which I'll thank you for sending us. And it says, do you think that there's a strong role for animal models when we can now use organoids? And if so, what types of animal models do you think are most promising? I presume that they mean for this particular type of tumor research, glioblastoma research, which is, we do have some papers on some of the reviews on animal models in Journal of Neuro-Oncology. So I would point you to that, but love to hear Dr. Lin or Dr. Emerson, your thoughts on organoid versus animal models.
Yeah, I mean, I think it's a great question. And I think, well, the issue is you have to decide what question it is you're trying to answer, right? So organoids are not meant to like replace animal models. They're supposed to be another arrow in the quiver for studying certain things. And I think what they're well suited for can be things like drug screening, invasion, microenvironment remodeling. That being said, these are not like intact organisms with functional vasculature and perfusion and things like that. They are the same species, so you don't have to worry about species mismatch and things like that. I think they're a little bit more tractable for me anyway than mouse models. Mouse models can be time consuming and expensive, but frankly, so can organoids too, if depending on how you do it. So I really think it depends on the question you want to answer, and you have to just acknowledge that old saying that, all models are wrong, but some are still useful. So.
Randy, thanks for joining, and Matt, Do you guys have any questions?
Yeah, sorry, and I apologize issues, but great talk. I was really excited to have you guys on today. And I think the question that I have, and it's really just insight from y'all, especially, since Chris, you do research on this also, is we're looking at, from what I understand, very precision medicine here. This is tailored not just to a person, it's tailored to parts of their tumor, even when you get into things like spatial heterogeneity and spatial transcriptomics. And my question is, how do you study this? Because we're dealing with a standard of care that is universally given right now to a variety of different tumors that, I think in the old days when I thought about this, I was hoping for like a Pied Piper approach. We could guide glioma down a route that we could keep treating it with a steady state of drugs, but we're not talking about that. We're talking about different diseases that react differently and respond differently. And how do we eventually come to conclusions about what works or what doesn't work with this tailored medicine? Because the numbers are going to be variable.
Yeah, and I think, if you really want to study this systematically, that's where you're going to run into the most problems, is just like, how are you going to have adequate power to really do rigorous comparisons, right? Especially if you're going to have a very refined view of things. So I think you've hit at one of the critical issues here. I think, as a first start, for the most part, I envision this as being whatever, we're able to hit upon with our studies, that it'll probably be something that's going to be adjuvant to current standard of care, not to replace, right? At least as a first step, I think that's the logical thing to do. Because, whatever criticisms you might have of our current protocol, it does provide some benefit. And I think those kind of strategies are more attractive for clinical trial design as well. So, lot of obstacles ahead, as you point out, but I think we can do some exciting stuff. Sorry, Matt, did you have something?
Yeah, no, I mean, this is really cool stuff. I mean, I think just to piggyback off of some of the comments from Dr. Sheehan and Dr. D'Amico, the question is, into Chris's earlier comment, looking at these classifications in the recurrent setting, can we use computer vision? Can we use better AI algorithms? Can we actually combine some of the clinical data that I think that you mentioned was kind of lacking in some of the, some of the data where some of these where some of your radiomics were kind of classified from, how we put that all together to make that actually tailored for the recurrent setting where some of these investigations in clinical trials could be moving forward. So it's more of a problem, and I think that we all have to kind of come together to solve. And so any thoughts about that would be welcome.
I think it's going to be an important part. It is also something we incorporated in our proposal. I think both you and Randy hit on some things that I think really apply both on the precision oncology front and the machine learning. And I'm always a little bit leery to lean into the current fads of whatever clinical practices, because I've seen a lot of those come and go throughout training, but I think they are truly important concepts. I think the most immediate application for us is, I have a collaborator here at the Hutch who's designed a machine learning algorithm to basically predict drug responses from a smaller subset of them. So you don't have time really to go and test 4,000 different compounds and all the various permutations and figure all that out. But by doing maybe a smaller amount from it, then you can extrapolate from that. So that gets you a little bit of efficiency. I know that's only maybe tangentially related to what you're asking, but I think gives an idea of the ways it might be applicable.
Well, that, or you find a crucial bottleneck, right? And we can start to target that through all these things. So, I think fantastic progress. It makes me excited about the future of things. I appreciate you being on. We're approaching the 29-minute mark here. So any last questions from the group?
No, thanks again. I think this is great work, and we look forward to seeing the next follow-up to this submitted to the Journal of Neuro-Oncology soon.
You got it.
Truly impressive work. Thanks for joining, and everyone have a happy Thanksgiving.
Yeah, you as well.
Thank you.
Thank you all. Appreciate it.
Happy Thanksgiving.